Welcome grade 12s. Today we are dealing with the topic called protein synthesis. What does protein synthesis mean? Let's look at the word synthesis. Where did you come across the word synthesis before? Yes, photosynthesis. What did photosynthesis mean? Let's break up the word. Photo means light. Synthesis means the making of or the manufacturing of. Photosynthesis means the manufacturing of food using light. Okay? So if we look at protein synthesis, once again, what does synthesis mean? Synthesis means the manufacturing of or the making of, in this case, a protein. So protein synthesis means the making or the manufacturing of a protein. And where is it taking place? It's taking place inside the cell. So protein synthesis is the process whereby proteins are manufactured inside living cells. Very good. Let's move on. There we go. So we see protein synthesis, it's the production of protein or the manufacturing or the making of proteins inside cells. But what are proteins? Proteins are polymers consisting of monomers called amino acids. Proteins are polymers. What does the term polymers mean? When we speak about a polymer, we are talking about a large molecule. So proteins are polymers, which are large molecules consisting of monomers. Monomers means building blocks. Okay? So the smallest block that a protein can be broken down into will be an amino acid. If I take a lot of amino acids and put them together, I form a protein. If I take a protein and break it down into its simplest form, into its building block, I would get an amino acid. So what are proteins? Proteins are polymers, which are large molecules consisting of monomers, which are building blocks called amino acids. Now, you might be thinking, why do cells make proteins? Well, proteins play a very important role in our bodies and it is necessary. So therefore it has to be made. Let's look at the functions of a protein and why is it so important to be made in the body? Well, we need to remember that excluding water and fat, the human body is made up almost entirely of protein. Now, protein is the main component, right, of our muscles, bones, our organs, our skin, our nails, our hair, okay? And excluding water, muscles are composed of about 80% of protein alone, making this nutrient especially important for athletes. Let's look at an example of an enzyme that's a protein, like our digestive enzymes such as amylase, lipase, pepsin, trypsin, okay? And the function of these digestive enzymes is it helps in the digestion of our food, okay? Another type of enzyme, which is a protein, is a transport enzyme like hemoglobin, albumin, okay? These transport enzymes help to carry substances in the blood, right? Or in the lymph throughout the body. Hemoglobin is a protein that helps to transport oxygen in our blood. Hormones are also proteins. Hormones are usually small proteins, right? Secreted by our endocrine cells. And they act to control or regulate specific physiological processes like our growth, development, metabolism, and reproduction. For example, insulin is a protein hormone that helps to regulate the blood glucose level. Let's move on. Let's recap the types of RNA. Can you remember where the RNA is located? Very good. RNA occurs in the nucleus as well as in the cytoplasm. It also forms part of the ribosomes. RNA consists of a single strand, also a polymer, which is a large molecule made up of nucleotides, which are their building blocks or their monomers. Let's recap. There are 3 types of RNA, namely the mRNA, which is the messenger RNA, tRNA, which is the transfer RNA, as well as the rRNA, which is the ribosomal RNA. The messenger RNA is found in the nucleus of the cell, but later moves out of the nucleus and attaches to the ribosome in the cytoplasm. Right? The transfer RNA is found in the cytoplasm of the cell, while the ribosomal RNA is found, as the name suggests, on the ribosomes in the cytoplasm of the cell. What do you see here? Yes, it is a structure of a cell. It's a cell. Okay, here's our cell membrane, cytoplasm, and our nucleus. So the first phase of protein synthesis takes place inside the nucleus, and it's known as transcription. What happens during transcription? It involves the formation of the messenger RNA. During transcription, the DNA double helix now is going to unwind, as you see in this picture. How does that happen? The weak hydrogen bonds of DNA break, forming 2 single strands of DNA. So now one strand, can you see the 2 strands separated? One of the strands is going to act as a template onto which a whole new strand is going to be built, forming a complementary strand. This complementary strand that's going to be formed is called our messenger RNA. Okay, a little bit of background. Now the DNA is sitting in the nucleus. The DNA can be compared to a reference book in the library. The DNA contains all the information on how to make a protein. The ribosome can be compared to a chef. He is the one that's going to be responsible in making the protein. But the ribosome needs the recipe on how to make a protein. The DNA is like a reference book in the library. It cannot leave the nucleus and come out, enter the cytoplasm and give this information to the ribosome so that ribosome can make the protein. The DNA cannot do that. So we need a middleman. The middleman we need is going to be our messenger RNA. We need our messenger RNA to take the information from the DNA on how to make a protein, leave the nucleus with that information and give it to the ribosome so that ribosome can now make the protein. So why is transcription important? Because transcription involves the making of our messenger RNA so that the process of protein synthesis can then take place. Let's begin with transcription. The making of our messenger RNA. What are the steps that are required for the making of the messenger RNA? Step 1. The DNA double helix needs to unwind, right? When the DNA double helix unwinds, the weak hydrogen bonds of the DNA is going to break. When that happens, it forms 2 single strands of DNA. One strand of the DNA is going to act as a template to form the complementary strand, which is now our messenger RNA. How is the messenger RNA formed? using free RNA nucleotides from the nucleoplasm. This process is called transcription. The next step that takes place is our new strand, which is our messenger RNA, needs to leave the nucleus, right? So step 2 involves the movement of the messenger RNA out of the nucleus through the nucleopore and into the cytoplasm, right? Where it's going to go and attach itself onto our ribosome. The 3 adjacent bases on the messenger RNA make up a codon. This codon is going to code for an amino acid. The next phase is called translation, which is taking place in the cytoplasm, and it's where the ribosome is using the information now from the messenger RNA to form a protein. What are the steps involved? According to the codons on messenger RNA, transfer RNA molecules now with matching or complementary anticodons will bring the required amino acid to the ribosome, as you see in this picture. Now, translation happens in the cytoplasm, right? What happens? RNA is translated into proteins by structures attached to the ribosomes. In the cytoplasm, we find the transfer RNA with its anticodons. The transfer RNA is going to pick up free-floating amino acids. The transfer RNA now is going to approach the messenger RNA with its anticodons. The anticodons on the transfer RNA is going to match the bases on the codons of the messenger RNA. The amino acids carried by the transfer RNA is now going to become attached by peptide bonds to form the required protein. Each transfer RNA is released to pick up more amino acids. Can you see? It releases itself to go and pick up more amino acids. As you have seen, the third phase of protein synthesis is translation. This involves free amino acids in the cytoplasm linking together in a sequence determined by the messenger RNA sequence. This process is controlled by enzymes. What happened during protein synthesis? To begin, we started with the process of transcription that occurred in the nucleus. Step 1, what happened? The DNA double helix unwinds and one of the 2 strands acts as a template. Free messenger RNA nucleotides in the nucleoplasm will bond with the complementary bases on the DNA template. Completed messenger RNA strand breaks loose from the DNA and leaves the nucleus. Then we have translation that takes place in the cytoplasm. Step 1, the messenger RNA now attaches itself onto the ribosome. So if you look at my picture, the green structure there is our ribosome. The messenger RNA is attaching itself onto the ribosome. The free amino acids are picked up by the transfer RNA. So here's our transfer RNA picking up the amino acids, okay, and is now heading down towards the ribosome, exposing its anticodons. Can you see the 3 anticodons on our transfer RNA? Right? They're going to be pairing with the complementary codons on our messenger RNA. Number 4, the amino acids now are going to bond. Here's our amino acids, these pink structures here. They will then bond into a chain of amino acids to form a protein. Please take note, in exam papers, different diagrams will be used, so please study your concepts and your labels. Do not memorize your pictures. Let's look at our structure here. Which phase of protein synthesis is occurring in this picture? Is it transcription or is it translation? Very good. It is translation. How do we know? We see the ribosome. Yes, number 1. Number 2, we see our messenger RNA with her codons. Very good. We see our transfer RNA with the matching anticodons. Let's look at our next structure. In this picture, what do we see? What process is taking place here? Very good. Here we see our double helix, our DNA with its double helix unwinding to form 2 strands. One strand acting as a template onto which a whole new strand is built. The new strand is our messenger RNA, and what's happening to it? It is leaving the nucleus. So this picture represents our transcription. The picture below, what do we see taking place here? Yes, translation, because again we see our ribosome with our messenger RNA attached to it. What do we see? Yes, according to the codons on the messenger RNA, the transfer RNA molecules are going to come with their matching or complementary anticodons to bring the required amino acid to the ribosomes. So that process illustrates translation. Very good. So please note, don't memorize. Understand what is happening in each picture to give you a clue.